Hey, that's my cue. Welcome, everyone. Good morning. Welcome to the Harmful Algal Bloom webinar. My name is Amy Weckel, and I'm from the Illinois Water Resources Center located at the University of Illinois, Urbana-Champaign, and I'll be your moderator today. This webinar is hosted by the Algal Bloom Action Team, of which the Illinois Water Resources Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and the University Extension within each state in the North Central region. In addition to hosting a webinar series, our team is developing a website of resources, including fact sheets and frequently asked questions on HABs topics. I encourage each of you to visit our website, which I'm hoping Ann can drop in the chat for us. Otherwise, I'll do it after I finish opening here. I encourage, at our website, you can explore some of the resources we have to date, including a What You Should Know fact sheet, a HABs frequently asked questions database, recordings from previous webinars, and our virtual HABs research symposium, which was held in January. Before we get started, just a few housekeeping items. We will have a Q&A session after our speakers present today. Please list your questions for our speaker in the Q&A panel, and we will do our best to address all of them following the presentation. If there's a question already posted to the Q&A panel and you have the same question, please upvote the question and we'll be sure to get the answers to the most popular questions during the session. Today's webinar is being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you're having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well, and we'll be happy to assist you as best we can. Today's presentation is titled HAB Effects on Wildlife and the Importance of Community Science. We will begin with a presentation by Dr. Janine Refsnider from the University of Toledo. Dr. Refsnider is originally from Minneapolis, Minnesota. She was an undergraduate with a biology major at the University of Minnesota Morris, got her master's in conservation biology at the University of Minnesota Twin Cities, and earned her PhD in ecology and evolutionary biology at Iowa State University. She was also a Fulbright graduate fellow at Victoria University of Wellington in New Zealand and a National Science Foundation postdoc fellow at the University of California, Berkeley. Dr. Refsnider has been at the University of Toledo since 2015. Her research focuses on reptiles' ability to compensate for the effects of climate change using behavioral plasticity, which she has studied in turtles, lizards, and tuatara. Her lab also studies how wildlife health is impacted by human-caused environmental changes, including climate change, chemical spills, and harmful outcomes. And I will now turn the sharing over to Janine.
Right, let's see. All right, everybody seeing my screen?
Yes, it looks great.
Okay, perfect. All right, so I'm going to talk today about some work that my lab has done on the effects of our harmful algal blooms in Lake Erie on wildlife health. So Lake Erie is pretty notorious for having harmful algal blooms mainly caused by microcystin algae really every year from late summer into early fall. So you can see Toledo is located kind of here at the very western edge of Lake Erie. And these harmful algal blooms that we get in Lake Erie are able to be seen from space. And when the algal bloom kind of makes its way toward our water intake structure, which happened kind of infamously in 2014, we end up with these water crises where about 500,000 people in the Toledo area were without drinking water for 3 or 4 days because dangerously high levels of microcystin were detected in the Toledo drinking water. And so since this algal bloom crisis in 2014, there's been a lot of effort into trying to figure out how these blooms happen, how to minimize them, how to improve our detectability so that we're less likely to have these water crises in the future. But there hasn't been a lot of research on the impacts of health in wildlife. So we know for humans that microcystin, which is the algal toxin that's primarily produced in the algal blooms in Lake Erie, microcystin is a liver toxin. So it's toxic if it's ingested. It can kill people. It has killed pets and livestock. We also know that if microcystin becomes aerosolized, such as in waves or boat wakes, it can also be toxic if it's inhaled. And potentially it's actually more toxic if it's inhaled from aerosolized particles than if it's ingested by drinking. And we also know that these health impacts on humans are exacerbated, particularly in people with pre-existing conditions. So people with liver disease, inflammatory bowel disease, and now potentially asthma are more likely to be seriously sickened from ingesting microcystin than people without these pre-existing conditions. But again, we don't know very much about the impacts of these algal blooms on wildlife. So very little is known. It's probably not good for wildlife. There was a correlational study in Lake Taihu in China, which has algal blooms caused by the same algae that we have here in Lake Erie. And in that study, there were quite a few dead species of wildlife washed up on beaches. And it was found that many of those species, including fish, ducks, and turtles, had high concentrations of microcystin in their tissue. We couldn't directly say that that wildlife was killed by the microcystin, so it's a correlational link only, but it is kind of concerning. And so my lab kind of set out to try to do some actual experiments and determine if there are impacts on wildlife health. And we were particularly interested in sublethal impacts. So not so much does algal bloom toxin, does it directly cause death in wildlife, but we wanted to see if there were these sublethal impacts. So could we see potential impacts on organ structure and functioning? Are wildlife species that are exposed to harmful algal blooms increasing their physiological stress levels, or perhaps depressing their immune functioning? And then might we see patterns in some of these impacts across taxa? So I'm going to briefly talk about two studies today that we have conducted in my lab. And the first is a controlled laboratory experiment where we exposed bullfrog tadpoles to microcystin to determine whether there was actual organ damage. And then the second is a comparative field study where we looked at 4 species of wildlife that are associated with wetlands to determine whether there were differences in either stress levels or immune functioning in species of wildlife from harmful algal bloom impacted wetlands compared to controlled unexposed wetlands. So I'll talk about this controlled lab experiment first. And so again, we did this study using bullfrog tadpoles. Bullfrogs are a little bit unique among amphibians in our part of the country in that they remain tadpoles for 2 years. So anything that they're exposed to in the water, they're exposed to that for a long period of time, not just a few weeks or months. And importantly, they're exposed to whatever is in the water during a really sensitive life stage. So they undergo pretty substantial metamorphosis toward the end of their tadpole stage. And so they're very susceptible to any contaminants in the environment. So for this experiment, we captured naive bullfrog tadpoles from a very isolated control wetland that had never experienced any kind of algal bloom. And we divided them into 2 groups. So half of those tadpoles we put in pond water that we then added microcystin to a dilution of 1 microgram per liter. And just to kind of point out, 1 microgram per liter is the threshold at which the EPA issues no drinking advisories. So this is a relatively low concentration of microcystin compared to what you might actually find in a lake during a harmful algal bloom. So we had 10 tadpoles that were exposed to this concentration of microcystin, and then 10 control tadpoles that were just in pond water with no microcystin. And we kept the tadpoles in these treatments for 7 days. Following the 7-day experiment, we euthanized the tadpoles and extracted and sectioned their organs. And so what did we find here? So I'm going to show a couple figures, and on all of these figures, the left columns are the control tadpoles, and the right columns are the tadpoles that were exposed to the 1 microgram per liter of microcystin. So we'll look first at some sections from intestines of these tadpoles. So these are longitudinal sections from one intestinal wall to the other. And we measured the diameter of these intestines and found, here's a normal sort of looking intestine in a control tadpole. If you look at a tadpole from the microcystin exposed group, though, the intestines are quite a bit wider. So the diameter increased. And also the walls of intestines, if you kind of zoom in on this part here, that's what this is showing. The walls of intestines should have lots of folds. So they're kind of going in and out and in and out to increase the surface area available to absorb nutrients from the food. And so we measured the maximum fold heights in a bunch of sections of the intestines of both tadpoles. And you can see we have pretty substantial fold heights in these control tadpoles. But the fold heights from the tadpoles who were exposed to microcystin are substantially smaller. And you can really barely see that there are even folds left in these intestines. And so what that shows is an inflammatory response in the intestines of tadpoles who are exposed to microcystin. So their intestines have essentially been stretched and widened. And then we also looked at liver tissue from both sets of tadpoles. So this line here is the outline, the perimeter of a liver cell. And you can see that the outline of this cell is substantially it's a smaller perimeter than the outline of this kind of expanded liver cell in an exposed tadpole. And then we also looked at the number of liver cells that were essentially binucleated, meaning that they have two nuclei. And it's difficult to actually pick out from this picture, but the number of liver cells that contained two nuclei was substantially larger in the exposed tadpoles compared to the control tadpoles. And when you start seeing binucleated liver cells, that's an indication that liver damage is happening and the cells are trying to repair themselves. So these tadpoles were exposed to only 1 microgram per liter of microcystin and only for 7 days. And in that time period, we saw liver damage and intestinal inflammation. So clearly that inflammation in the intestine is probably limiting the ability of these tadpoles to get nutrients from their food. And that could easily lead to malnutrition if it occurs for a long enough period. And we didn't actually study this, but it's likely that these liver damage and intestinal inflammation responses that we saw are probably exacerbated in animals who have other pre-existing conditions. So we did see pretty substantial evidence for organ damage over a very short time frame in these tadpoles. Okay, so we did a second study, and this was more of a comparative study in the field, where we compared stress and immune functioning in animals that were commonly associated with wetlands from both harmful algal bloom exposed wetlands and control wetlands. And so we did this in 4 species, 2 species of avian reptiles or birds, barn swallows and red-winged blackbirds, which are very common. Both of these species live right over wetlands and they feed their babies insects that have just emerged from the wetland, and they also eat insects that are kind of coming right out of the wetland. And then we also did this study using 2 reptile species, northern water snakes and painted turtles, 2 other very common species. Both of these species spend a lot of time in the water and they eat food directly out of the water. So water snakes are primarily fish eaters. They spend a lot of time hanging kind of over the edge of wetlands and swimming in wetlands looking for fish. And painted turtles are primarily herbivores, but they eat aquatic vegetation and they spend most of their time in the water. So these are 4 very common species that spend most of their time in or eating out of wetlands. And we conducted this study at 2 different sites. So we had a control site at Ottawa National Wildlife Refuge, just outside of Toledo. And at this site, at the time that we did our study, there was no detectable microcystin in the water of these wetlands. So it was below the detectable limit of 0.03 micrograms per liter. So that again was our control site. And then our studies, our HABs exposed site was this lake called Grand Lake St. Mary's in Mercer County, Ohio. And this lake has been undergoing a chronic algal bloom, with microcystin being the primary algal toxin produced. It's been undergoing this algal bloom for about 2 years straight, including in the middle of winter. So this lake was man-made, it's extremely shallow, it doesn't undergo any normal turnover, and the entire watershed is agricultural, so there's a huge amount of nutrient input. And this lake looks like bright emerald green pretty much year-round. And the microcystin concentration at the time of our study was between 40 and 50 micrograms per liter. And just as kind of a benchmark, the EPA issues no contact with the water advisories at 20 micrograms per liter. So this lake was more than twice the concentration at which you shouldn't even come into contact with the water. So it's this really pretty lake with lots of houses and docks and everything around it, with nobody in the water and nobody even supposed to be really even along the edge of the water. But that doesn't mean that there isn't wildlife in the lake. So this again is Grand Lake St. Mary's, and there are turtles basking in the logs here. There's a whole family of geese. So there are lots of wildlife species just living in the lake as if it's completely normal. So we collected blood samples from animals of all 4 of those wildlife species at both the control site and this harmful algal bloom exposed site at Grand Lake St. Mary's. So we collected blood samples from all of these species, and we measured physiological stress levels using an index called the heterophil to lymphocyte ratio, which I'll explain in a minute here. And then we also measured immune functioning using 2 different assays. And for those, we collected blood samples in the field, separated them immediately. So here's the packed blood and the plasma, and we flash froze the plasma and then used that subsequently for some lab assays. Okay, so the first thing we did here again was to measure physiological stress levels using H/L ratios. And we did that by making a little blood smear on a slide, which we then would stain. And then under a microscope, we would look at the blood cells, and we were concentrating on the white blood cells. So there's essentially 5 different types of white blood cells involved in various aspects of an immune response. And we classified each of the blood cells, the white blood cells, into a specific type of these 5. So this is a slide, I forget which turtle species this one is, but turtles and birds are a little bit unusual compared to what most people are used to, in that their red blood cells are nucleated. So most of the cells that you're seeing in this picture are red blood cells. So we ignored those and we focused only on white blood cells. So we would count the first 100 white blood cells that we saw and classify each of them as one of these 5 types. And the ratio of these 2 specific types, so heterophils, which are kind of these globular things, compared to lymphocytes, which are these little purplish kind of amorphic structures, the ratio of heterophils to lymphocytes is an indicator of the stress level, the baseline stress level. And this is a more useful indicator of chronic stress in wildlife than something like glucocorticoids, because things like corticosterone and cortisone can increase within seconds of an animal being captured and handled. And so then you're essentially measuring the kind of stress response rather than the baseline stress level. So the H/L response takes days to increase following a stressor. So if you're just capturing an animal and collecting a blood sample, you're not confounding what you're measuring with the stress that you're inducing by handling the animal. So our stress measure again was this H/L ratio, where the higher the ratio, the higher the baseline stress level. And then we also conducted 2 assays on innate immune functioning. So again, we have multiple kind of lines of defense in our immune system, but your innate response is really the first line of defense against any kind of infectious agent. So it's relatively energetically cheap to mount, and it's non-specific. So it's not as specific and targeted as an immune response to something that you've experienced in the past, which is how vaccines work. But it is the response that kind of your body mounts immediately when you're confronted with a novel infectious agent. So we did 2 different metrics of innate immune functioning, and both of these are pretty commonly used in wildlife studies. So we measured the bactericidal capacity of each individual, and that's essentially the ability of complement protein in the blood plasma to kill bacteria. So basically what we did was to incubate E. coli colonies with plasma from each individual animal, and then grow the colonies on a plate. And we would compare the number of colonies that grew in a plate containing plasma to the number that grew in a plate containing no plasma, or a control plate. And so you can see that there are substantially fewer fewer colonies in this plate of plasma compared to the control plate. So this animal killed a pretty high proportion of the colonies that would otherwise have grown. So the higher your bactericidal capacity, the higher your immune functioning is. And then our other assay of innate immune functioning was the natural antibody agglutination ability, which is essentially the ability of your antibodies to kind of agglutinate to foreign cells. And in this case, we used foreign, I think it was rabbit red blood cells. So we basically combine plasma with foreign cells, in this case, rabbit red blood cells, and then see on this plate how dilute can the plasma sample be and still exhibit the ability to kill the foreign cells. So again, the higher the number for the agglutination ability, the higher the immune functioning for that animal. All right, so what did we find here? We found that the H to L ratios, so again, that stress level, was higher from animals in the harmful algal bloom exposed wetland compared to the control animals in 3 of the 4 species. So in both bird species and in the water snake species, there were higher baseline stress levels in the animals from the harmful algal bloom wetland compared to the control wetland. And there was no difference in the turtles. So that suggests that at least for the birds and the water snakes, the harmful algal blooms do present an environmental stressor. So those animals did have higher baseline stress levels because they were in a wetland that was chronically exposed to a harmful algal bloom. And we also know that in general, chronically elevated stress levels depress other important functions in your body, such as immune functioning, reproduction, digestion. And so we would then predict, because these 3 species had higher stress levels in the harmful algal bloom wetlands, we expected they would probably have lower immune functioning as well. But that actually wasn't what we found. So as far as the bactericidal capacity, we found no difference between the wetlands in either bird species. So in both species, bactericidal capacity really wasn't affected by the harmful algal bloom. We found that in water snakes, the snakes from the harmful algal bloom wetland actually had higher immune functioning than the snakes from the control wetland. And we found the reverse in the turtles, where the turtles from the harmful algal bloom wetland had lower immune functioning than the control turtles. So the turtles were really the only species that matched our prediction. And none of the species showed any difference between wetland sites in that other measure of immune functioning, the natural antibody agglutination. So we found here again that turtles in the harmful algal bloom wetlands had depressed immune functioning. So clearly this could make them more susceptible to things like diseases and parasites and could impact their health down the road. But in the water snakes, we found higher immune functioning in the harmful algal bloom wetland. So if you have higher immune functioning, if your immune functioning is ramped up for some reason, that's going to come from somewhere. That energy isn't free. So there could be costs because of these increasing immune functions in water snakes from the harmful algal bloom wetlands. And those costs could come from things like reproduction or body condition. So perhaps these snakes with higher immune functioning in the harmful algal bloom wetlands actually have lower reproductive output or worse body condition than snakes from the control wetland. So really what we're seeing is that there are a wide variety of impacts that these harmful algal blooms have on wildlife health. So we saw malnutrition evidence in the tadpoles. We found potential for decreasing body condition or reproductive output if there are costs associated with these impacts. And really, any of these impacts that we saw could have population level consequences. So we were seeing effects on individual health, but if a large population is all experiencing health impacts, that could lead to overall lower reproductive output in the population, potentially shorter lifespans and higher mortality overall, which could lead to population declines. And so this study is really important because it kind of shows a mechanistic understanding of how physiological responses of individuals to human-caused environmental changes could contribute to population level declines. And this is important because a couple of the species that we studied, even though they are very common, they have close relatives that are federally endangered. So for example, Lake Erie water snakes are very closely related to the common water snakes we studied. There are a few species of endangered turtles that are closely related to painted turtles. And so if these individuals in Lake Erie are having negative impacts on their health, if you're seeing that same thing in these very rare species, that could easily lead to population declines of these very endangered species that could be contributing to overall endangerment of those animals. So clearly the next steps here are to kind of look at whether there are population level impacts of these health impacts that we're seeing. Are there costs of this increasing immune functioning in water snakes? So are water snakes during harmful algal blooms having worse reproductive output or worse body condition? We also know that in reptiles, species can use behavior to adjust for environmental changes such as climate change. So my lab has found previously that, for example, turtles can change their choice of shade cover over nests to counteract effects of climate change on offspring development. We know that lizards can change their basking behavior if they're exposed to a warmer climate. So it's possible that these animals might be able to behaviorally kind of compensate for the effects of harmful algal blooms by perhaps actually avoiding them altogether and avoiding exposure to them. So we do have another study ongoing now where we have satellite transmitters on some turtles in Lake Erie to try to track their movements and map those onto the movements of an algal bloom to see if the turtles are behaviorally avoiding algal blooms and therefore not even becoming exposed to them. So I think that's it. I have a lot of people to thank, and I'm happy to take questions. I guess we'll wait till the end for those though.
Thank you so much, Janine. That was really interesting. Yeah, we will do questions at the end. And so now I will share my screen again and we will go to our second presentation. Our second presentation is by Dr. Algerich. I'm sure I messed that up. From the University of Missouri. Alba is an assistant professor at the School of Natural Resources at the University of Missouri. Her research group studies the effects of land use on water quality and ecosystem integrity using experimental and modeling approaches. Among other projects, she leads the Lakes of Missouri Volunteer Program. This community-based science project has monitored water quality in Missouri reservoirs for more than 30 years. And Alba, you can go ahead and take it away.
Thank you. So let me see if I can share the screen. Are you seeing my screen now in presentation mode?
Yes, it looks great. Thank you.
Perfect. Excellent. So thank you very much for giving me the opportunity to talk about my favorite project, the Lake of Missouri Volunteer Program. My name is Argerich. I'm an assistant professor at the School of Natural Resources. And today I'm going to talk about this citizen science project, this community science project that has been going on for more than 30 years. It started in 1992 by Dr. Jack Jones at the Immunology Lab at the University of Missouri. Since then, it has been run by different project managers, including Dan Abrett, and right now currently run by Tony Thorpe. He has been running the program for 20 years now, more than 20 years. And the goals of this community science project, it's basically understanding current lake water quality, monitor for long-term changes in water quality, and importantly, educate the public about water quality issues. Throughout all these years, we have kept our sampling and lab protocols pretty constant. Anytime that we had to do a change because of new techniques or new technologies available, we have made sure to have a consistency in the data. So right now what we have is more than 30 years of data that running, right, for the Missouri reservoirs, which is high quality. And I talk about community science, but well, I'm sure that all of you are familiar with it. But as California Academy of Sciences define it, it's a global movement through which scientists and non-scientists get together to make observations, collect data, and help answer some of our planet's most pressing questions. In Missouri, one of the pressing questions is water quality. Importantly, I think that what makes this program so special, it's basically a grassroots movement. It's where people that are not scientists get together with scientists to respond to these water quality concerns. Volunteers know and love their sites, so they are good stewards. They try to make the right decisions, and they are vigilant about any changes that they are seeing. And because they live usually close to the site, so we are able to have rapid responses to sudden quality changes. So usually when something like that happens, they alert us. We are able to send some sample kits, and we're able to detect some changes or do some analysis. And then we connect them with MDNR or any state agencies so they can help them solve the problems, right? In addition to that, and this lovely picture here on the bottom corner shows it. This is a family business. So volunteers are not volunteering alone. Usually they go with friends, with families, and it's another way of educating, right? And creating this stewardship movement. The success of this volunteer program, these 30 years, basically it's having good partnerships. So the program it's funded by MDNR, the Missouri Department of Natural Resources, through EPA money. We have also good partnership with the Missouri Department of Health and Senior Services, who provides kits for toxin analysis. For the volunteers basically agree on picking up a lake that they are willing to monitor multiple times throughout the summer, usually 8 times between April and September. They need to have some equipment like a boat or any access to ways of sampling, but we will provide all the supplies needed for that. They then go to the field and they measure, they collect the samples, they do some processing at home, and then they send the samples back to our lab where we analyze them. Our responsibilities as a program, basically, we have personalized training. So we're a little bit picky about the volunteers that we're accepting the program. We want to make sure that we're investing time in them and that they are willing to do the work afterwards. We have one-on-one personalized trainings. We provide the supplies, and what we do is a lot of follow-up, right? So the volunteers go to the site during the summer. They send the samples back. Then we do the analysis, and we respond to them, right, with the results of those analyses. Right now, we have 150 active volunteers, which is pretty awesome. We have high retention. Some of the volunteers have been sampling for 15 years straight. And altogether, this creates high data quality, right? Because of these personalized trainings, the follow-up, the trainings are happening not only at the beginning of when a new volunteer arrives, but we have multiple checkpoints throughout the sampling life of this volunteer. This produces high data quality that has been used in multiple scientific publications. In addition to that, what we do is we have a web page, an active web page, where we post different topics about water quality. We produce 2-3 newsletters per year on the Waterline. You are able to subscribe even if you are not a volunteer. We have presence in social media, and then we provide public talks, outreach events. We went to the library last year, here the public library. We go to homeowners associations if they ask us to talk about their lakes and the health of their lakes. And this is how it has grown. So what you're seeing here is the number of sampling sites starting in 1992 to 2022. Those are the columns. The red line shows the number of lakes. So usually we have, minimum we have 1 sampling site per lake at the dam site. But some of the largest lakes have multiple sampling sites. And I'm talking about lakes, but in fact, what we have in Missouri, it's basically reservoirs, so they are all man-made. So this is why I'm saying that we're sampling by the dam. Our surface area can range between 21,000 hectares, Truman Lake, Lake of the Ozarks, to 4 hectares, Robbie Lake. So we have a wide range of surface area of those lakes. The median is 57 hectares, so it's a medium size, small size. Estimated depth ranges between 5 and 76 meters with a median of 15 meters. And in general, again, so we sample at the dam. It's not targeted sampling, so they go in regular intervals. And in our largest sites, right, like Table Rock here, we have multiple sampling sites that are run by different volunteers. Here is a picture of sampling sites throughout the life of LMVP, of the Lake of Missouri Volunteer Program in light blue. And here on the upper right corner, you see this map of Missouri. So basically, Missouri has two clear ecoregions. The northern part, it's the plains ecoregion. The lakes here are usually more turbid. It's where most of the population of Missouri lives, and it's mostly flat agricultural, right? So the water quality issues are usually more extreme. The green region, it's the Ozarks region. It's more of a hilly region, forested, water quality. The water quality usually it's better, although we have had in this area, we don't have as much agriculture, but we have a lot of mining legacies. So we had a lot of mines going on in the area. But in general, water quality for the parameters that we're looking at, it's in general better. Our sampling sites are a little bit skewed, right? Representing where the population is. So 70% of our sampling sites are mostly in the plains and 40% in the Ozarks. And this is how the workflow looks like. So basically, January, March, it's our prepping time. We create labels, we prepare bottles, we prepare the supplies that we're going to send to the volunteers. Even during the pandemic, right here you see a nice picture of Tony, our project manager, preparing sampling supplies that want to be sent to our volunteers. He was working from home at that time. Then between April and September, the volunteers sample every 3 weeks, resulting in 8 sampling events throughout the season. They go, they collect the water, and then they come back at home where they process this water. They filter it, they preserve it, and they keep them, they keep the samples frozen at home in their refrigerator, in their fridges. Between June and December, Tony and other lab staff goes around Missouri collecting samples from the volunteers and bringing them back to the lab, where we start doing lab analysis and data end. And finally, by December, usually we're done. January, March is reporting time, where we prepare the lake report for the previous year. So which parameters we monitor? Well, we have water temperature, water clarity, chlorophyll, nitrogen, phosphorus, and suspended sediment. With water temperature right now, where some of our volunteers are experimenting with fish hawks, so they are able to get profiles of water temperature to understand a little bit better stratification. But in general, most of the samples are collecting at the surface. And those are the results for the 2022 campaign. So what you're seeing here are the results for water clarity, which we measure with a Secchi disk. You can see in red are Secchi depths that are very shallow. So turbid places, basically, in blue, real clear lakes, and in black in between, right? So you can tell that basically most of the highest turbidity it's in the plains region, which would be expected because of the type of geology that we have and the fertilization issues that we have in the area. The Ozarks region, it's usually where you find the clearest water. And chlorophyll concentrations, they vary across the ecoregion. So the threshold, this separation is not as clear. What we're seeing here again, it's in red, high chlorophyll concentrations, in blue, low chlorophyll concentrations, in black, in between, from 2022. Our average chlorophyll concentration, it's around 19 micrograms per liter this year. When you look at the long-term data set, this average, usually it's around 21. So this year we're a little below of the total average that we have seen. Chlorophyll concentrations, in fact, respond better to phosphorus concentrations, so more than Secchi disk or clarity. So here you're seeing phosphorus concentrations across the sampling sites in 2022. In average, we have around 44 micrograms per liter phosphorus. This is total phosphorus. The range, though, it's huge, right? It ranges from 3 to 500 micrograms per liter. And again, this year we were a little bit below the average for the total, from the total data set that we have. So we had 44, and the total average it's 58. And again, so this chlorophyll mostly responds to phosphorus concentrations. We're able to see this nice relationship, and this is how we base our trophic states. So I will show you in a second how our trophic state changed based on these total phosphorus concentrations. But before, let me talk about a little bit about nitrogen concentrations. Nitrogen concentrations are one order of magnitude higher than phosphorus concentrations. Mostly centered into the plains region. Also, we see some inputs also in the lowest corner of the state. And basically, again, this relationship between nitrogen and phosphorus is not really linear. Based on the trophic state, what we're seeing basically is that here what you are seeing the number of sampling sites that we have on the vertical axis across years. Split between trophic state based on these total phosphorus concentrations. What you can tell is that our sites are mostly eutrophic. This is our dominant trophic state. We have some hypertrophic lakes and very little oligotrophic, which might respond to also to the efforts, right, that we are putting into where the volunteers live, where they are, where these oligotrophic lakes are. But basically, what we are seeing is this general dominance of eutrophic conditions. Let's talk about toxins. We are also sampling for toxins. This toxin analysis, toxin monitoring started in 2015 for microcystin. 2017, we started with cylindrospermopsin. Again, this is largely ambient monitoring. We're not targeting algal blooms. So what we're going to see here is basically what occurs right throughout the summer without any target algal bloom monitoring. We're always sampling at the same spot, so we're able to track changes in time. And the volunteers, what they are doing is they are collecting an integrated sampler. Here you can see a picture of the sampler itself. It's a tube that it's 45 centimeters long. Basically, that collects water throughout the whole column, and we replicate that 3 times, and then we composite the sample. And then the volunteers collect this sample, and they preserve it, and they freeze them until it's sent back to us for analysis. So this is what we have seen for microcystin in 2022. Basically, we have a couple of 3 sites that have shown high microcystin content. Most of them are above detection limit, and similar, it's happening with cylindrospermopsins here. So with cylindrospermopsin, we're seeing also a general trend. We see a lot of occurrence above detection limits. And if you're curious to know about how this looks like in terms of trends, what we're seeing here is basically that microcystins occurrence basically shows higher occurrence in hypertrophic sites, whereas cylindrospermopsin, though, we see an opposite pattern. So we see more cylindrospermopsin happening when in alloeutrophic lakes in comparison to hypertrophic lakes. However, when we look at the spatial, the geography, right, where are those percentage located? Basically, they are pretty similar. They are both in the plains where we're seeing more of those occurring. Across years, this is what we have seen. So here I just pulled data from years where we have both microcystin and cylindrospermopsin. This is the average toxin concentration that's seen at this year, compositing all the samples that we have for that year across all lakes. What we see, and both of them, both cylindrospermopsin and microcystin, have similar sample numbers. So you can see in the table below the sampling, the samples that we have for each year, for each of the parameters. What you can see is that, well, starting in 2017, we saw a dip in microcystin concentration, but since then it just started rising again somehow. Cylindrospermopsin, though, it's still varying. Okay, So summarizing, all this data that we're collecting, it's used to understand trends in water quality, informs MDNR project, MDNR and EPA when they are trying to understand guidelines and to assess water quality across Missouri. In terms of toxins, we see opposing appearance in terms of trophic status, but similar regional distribution across microcystin and cylindrospermopsin. And in general, that this program is quite unique because of the high volunteer retention rates and data quality. We think that the case to success after looking at all these years of data and this experience is basically those personalized trainings and follow up after all these efforts that are made, right? The sampling and freezing the samples and then sending the samples back. And especially having dedicated personnel and partnerships. Those are key to success. So right now we have 3 people dedicated exclusively to this project, 1 project manager, and then 2 persons in the lab that are able to analyze and process all these samples. And with that, if you would like to know more, just please visit our webpage, send us an email, sign up for our newsletter, and I will be happy to take questions.
Thank you so much, Alba. That was also very interesting. And we do have quite a few questions. First, let me share my screen one more time. Okay, I'd like to thank everyone for participating in the Algal Bloom Action Team's webinar series. A special thanks to our presenters for sharing their research with us. And also thank you to Ann Nardi and Joe Bonnell and their team at the North Central Region Water Network for hosting the Zoom webinar and coordinating the technical aspects of the webinar. We couldn't do it without them. Following the Q&A session, please complete the survey at the link that I'm providing here. 1 second. I'll put it in the chat as we get through going through the Q&A. We appreciate any additional comments or feedback you have on our series. And we'll now begin the Q&A session. We will end right at noon. And so if there are questions that we don't get to, feel free to reach out to the speakers themselves. Their emails are provided on this slide. And hopefully they can answer your questions for you. So the question with the most upvotes so far is from Lois Wolfson. Are there any plans, and I believe this is for Janine, are there any plans to look at exposed tadpoles into their adult stages and examining more long-term effects of the microcystin exposure?
That's a great question. I had a PhD student kind of interested in really looking at impacts on hibernation because the livers also, in many species that hibernate of amphibians, the livers produce kind of almost antifreeze proteins that allow these frogs to, in some cases, actually freeze underground. It prevents ice crystals from forming in the cells. And obviously, if there's liver damage, that could impact the ability of these species to hibernate. And we have not pursued that, although it is something that we're interested in. I've also found in the last couple of years that impacts of harmful algal bloom toxins on wildlife is becoming a lower funding priority. And so it's getting harder to get some of this stuff funded. What they're really interested in is preventing the algal blooms from starting in the first place, which makes sense. So we're kind of fighting for funding to get the ability to do that kind of thing. But that's a great question. That's something I'd really like to pursue.
Great, thank you. The next question is, this is also for you, Janine. How did you apply microcystin in the first study as a raw chemical or by introducing microcystis cells to the tadpoles? And do you think that there might be a difference between the two?
We collected algae from a harmful algal bloom in Lake Erie, and we diluted that down, and that was what we introduced. So it wasn't a raw chemical, it was the actual algal cells from the lake. It's possible that there would be a difference because what we introduced was essentially the full component of microcystin, which included intracellular and extracellular. And I could see that there might be a difference if you had just like a chemical by itself, that it might only include one of those. we didn't test for a difference between types of microcystin. We just literally pulled some out of a lake, quantified the concentration, and then diluted that down, and that's what we added.
Okay, great. Erin, I hope that answered your question. The next question comes from Madeline. And could other stressors related to HAB formation, such as temperature or water quality parameters and presence of other freshwater parasites or diseases, contribute to that baseline stress level being higher in the HAB wetland?
Yeah, it definitely could. And there's so much agriculture around, especially Grand Lake St. Mary's, that there is almost an infinite number of things that could be in the water that we didn't quantify. So this is really just correlational. We would have to do a similar controlled lab experiment, like what we did with the tadpoles, but do that with these other species to really add a causal link to what is contributing directly to that stress level that we measured. So it's definitely possible that there's other stuff out there that we didn't look at, but we didn't attempt to measure everything.
Okay, great. For Elba, where Caroline lives, the state cautions against citizens sampling microcystin because of the health concerns. How did Missouri decide the benefits of using volunteers to collect microcystin outweighed the risks?
Good question. In general, so they are, because it's observational sampling, it's not targeted. We are not sampling during algal blooms. I think that makes a difference. Volunteers are cautioned about being away, right? Not exposing pets and everything. If they see an algal bloom, an active algal bloom, they report it basically to MDNR or basically MDNR, so they are able to access the site. So this is what we try to avoid, right? Active algal blooms, basically.
Great, thank you. Janine, another one for you. In your second study, how do you separate the effects of microcystin from all the other stressor variables associated with causes and effects of HABs?
Yeah, so we really couldn't. It was correlational only. So we had, that was the main thing that we measured, but there are potentially lots of other chemicals or contaminants in the lake that could have contributed that we didn't control for. So we would have to do a controlled lab experiment, an exposure experiment, to separate out, things like, fertilizers, for example, specifically, as opposed to microcystin.
Okay. One more for you, Alba. Can you please talk more about how the volunteer monitoring is funded? Does the state have grants from US EPA?
Yeah, that's correct. So basically it's MDNR funnel EPA money, right? So through from MDNR. We have had some extra, the Missouri Health Department, it's helping out with supplies, but mostly it's MDNR money.
Great. And Janine, do you have plans for exposing tadpoles and then tracking their health and fitness through metamorphosis? And how might adult frogs who were exposed as tadpoles be impacted?
Again, my prediction would be that it would impact hibernation and survival during hibernation, because the same organ that is damaged by microcystin also produces chemicals that help the frogs to survive hibernation and survive freezing. So I would expect that it would lower winter survival as opposed to anything else. We don't at the moment have plans to study that, but that would be worthwhile in the future.
Okay. And Patrick is wondering, how did you ensure there were no harmful algal blooms in the control wetlands throughout the course of the field study? These blooms can be temporarily variable. Did you continue to sample for microcystin during the study?
So we only sampled animals over like a 5-day period each year, and we did measure water microcystin concentration in the water during that 5-day period. And so the numbers that I reported for microcystin concentration were from that same short time that we sampled animals, and in the control wetland, it was undetectable, at least for those 5 days. But we've also measured microcystin concentrations in that same wetland at other times of year and have never detected detectable levels.
And if you've mentioned this before, apologies, but which microcystin container did you use for exposure and find in the environment?
We were measuring for MCLR, and I think it's been a couple of years. I think we also measured for MCRR, but MCLR is by far the predominant congener in the harmful algal blooms in Lake Erie, and that was what our ELISA assay focused on.
Okay. Also related to amphibians in wetlands, have you seen impacts of microcystin on the success of amphibian eggs, such as spotted salamanders and toad or frog eggs?
We really haven't looked at impacts on eggs at all, and there aren't salamanders at either of the sites that we targeted here, so I can't speak to salamanders at all. We did have a really hard time. We actually were planning to do this field study with bullfrog tadpoles as well. And the site that had our microcystin exposed site, we had such a terrible time even finding bullfrogs to sample that we ended up not being able to include them. So just anecdotally, I think it has a pretty severe impact on at least frog eggs and tadpoles, that there just aren't any to be sampled. So I would expect that that's probably the case for salamanders as well. But again, there aren't salamanders at these exact sites.
Okay. And Alba, do you conduct audits of the volunteer teams where you take side-by-side duplicate samples?
Absolutely, yeah. We do that regularly. So we double check that the process are accurate, and we do a split sampling sometimes. So we have staff sampling side by side with the volunteers, bringing the samples back to the lab and comparing results. And if we see some discrepancies, then we do the retraining, right? We train again. So each volunteer has kind of a check
Okay.
for the quality.
Great. And do you know the demographics of your volunteers, and where do you recruit them from?
The demographics, it's a little bit all over the place, but we have a lot of retired people that they have the time, I guess, and the money, and they live side by side the lakes. So, but again, because it's a family business, right? They bring children with them. They can bring the grandsons or the grandchildren. The recruitment happens mostly word of mouth, and then during these outreach events that we participate on. So we have we participate in the Earth Day maybe, or the library organizes talks, right? That's when it's happening. We give talks in the classroom too. Sometimes we get student volunteers, so a little bit of everything.
There's one more here and it just jumped away from me. So I'll go back to this one. For Janine, is the intestinal and or liver damage sustained enough to prevent the tadpoles from maturing to adulthood?
That's a good question. We didn't follow them long enough to tell. I would imagine that if they were maintained in that level of microcystin, that would probably impact them. I would think it would prevent them from reaching metamorphosis. They would essentially not survive. But we didn't keep them in there long enough to tell. So that would be worth testing, but I can't say for sure.
Okay, Alba, have you looked into the opposite trends in microcystins, cylindrous omoxin, that's a tough one for me, versus eutrophication? Have you come up with any potential explanations?
No, but yeah, we have several ideas, right? Could be some type of exclusion, competitism between one and the other. But right now we're exploring the data. I think it's pretty cool, but I don't understand yet what's going on there.
Okay. Let's see. It says our local lake management groups have been using chemicals to control weeds interfering with recreation users, not very effectively. Is there data on wildlife effects on treated lakes? Either of you?
Yeah, I'm not aware of anything. I don't know.
Sorry, could you repeat the question? I was trying to look into the QC.
Yeah, local lake management groups have been using chemicals to control weeds interfering with recreation users. Is there data on wildlife effects on treated lakes that you know of?
I know that they are doing quite a bit of research on waterfowl. Here are some of our research colleagues in the university. But yeah, we're not doing anything like that in our group.
Okay, and one final question for Janine before we wrap up here. Why bullfrogs rather than another amphibian?
Bullfrogs are really large, and so that makes them very easy to get a blood sample from without hurting the bullfrog, which was kind of our original design. And they're also really easy to keep in captivity. So that was really it. They're very common. They live as tadpoles for a long period, so that makes them a good model species for kind of understanding impacts versus like toads that are only tadpoles for a week or two. So that was it.
Great, thank you. And there are still some really great questions remaining. If the speakers didn't get a chance to get to them, please feel free to reach out to them at their email addresses that are on the screen. And yes, the slides, I believe from both speakers' presentations, as well as the recording of this webinar, will be uploaded to our website at the Algal Bloom Action Team website through the North Central Region Water Network. And again, thanks everybody for participating, and we'll see you again in a couple of months. Take care.